Rosemary. There is a village on the Italian coast called Acciaroli. Around 300 of its 2,000 residents are over 100 years old. That is one of the highest concentrations of centenarians anywhere on Earth. Researchers from the University of California, San Diego, and the University of Rome studied the village in 2016. They were looking for what made Acciaroli different. The diet was Mediterranean, the lifestyle was active, and rosemary was used in almost every meal every day by almost every resident. Rosemary contains a compound called carnosic acid. Laboratory research has found that carnosic acid activates a pathway in the brain that protects neurons from oxidative damage, the kind of damage associated with cognitive decline and Alzheimer's disease. A 2016 study in Scientific Reports identified the specific mechanism by which carnosic acid protects brain cells under stress. A separate study published in Therapeutic Advances in Psychopharmacology found that simply being in a room diffused with rosemary aroma improved speed and accuracy on cognitive tests. The compound responsible, 1,8-cineol, was measured in participants' bloodstreams after inhalation and correlated directly with test performance. Rosemary has been in every kitchen in Britain and the Mediterranean for centuries. It grows easily, costs almost nothing, and is available everywhere. The science behind why Acciaroli's residents live so long is not fully understood, but rosemary is consistently present in every account of their daily lives. A herb already in most kitchens, with brain protection research behind it, and a village of centenarians who have been proving the point for generations. Bacopa. Bacopa monnieri has been used in Ayurvedic medicine for memory and cognitive function since at least 300 BC. Children in India were given it before examinations for centuries. Modern clinical trials have now tested exactly what ancient practitioners observed. Bacopa is a small creeping plant that grows in wetlands across South Asia. The active compounds, bacosides, appear to support the repair and regeneration of nerve synapses, the connections between brain cells that degrade with age and stress. A 2001 double-blind randomized controlled trial published in Psychopharmacology found that Bacopa extract significantly improved memory acquisition and retention in healthy adults over 12 weeks compared to placebo. A 2008 study in the Journal of Alternative and Complementary Medicine found similar results in older adults specifically, the group where cognitive aging is most relevant. The mechanism is specific. Bacosides appear to reduce the breakdown of acetylcholine, the neurotransmitter most directly associated with memory and learning, and to reduce oxidative damage in the hippocampus, the brain region most affected by age-related cognitive decline. Bacopa is not a stimulant. It does not produce immediate effects. Clinical studies consistently found that benefits emerged gradually over 8 to 12 weeks of daily use. That timeline matches exactly how Ayurvedic practitioners always described it, a tonic taken consistently, not a quick fix. 3,000 years of use for exactly the same purpose it is now being validated for. The people who used it before the clinical trials were not wrong. Amla. The USDA tested the antioxidant capacity of hundreds of foods using a measurement called ORAC, Oxygen Radical Absorbance Capacity. Amla, the Indian gooseberry, scored higher than almost anything else they tested, including blueberries, pomegranates, and acai. Phyllanthus emblica has been used in Ayurvedic medicine for 5,000 years. It is one of the 3 fruits in Triphala, the foundational Ayurvedic formula used as a general health tonic across South Asia for millennia. In Sanskrit texts, amla is described as a rasayana, a rejuvenating substance that promotes longevity. The antioxidant activity comes from an unusual combination of compounds including emblycinin A and B, tannins found almost exclusively in amla, along with high vitamin C content that remains stable even when the fruit is dried and heated, unlike vitamin C in most other fruits, which degrades rapidly with processing. Modern research has looked at amla specifically for effects on aging markers. A 2011 study in the European Journal of Clinical Nutrition found that amla extract reduced markers of oxidative stress and improved cholesterol profiles in healthy adults. A 2014 study in the journal Phytotherapy Research found anti-inflammatory effects comparable to pharmaceutical anti-inflammatory drugs at equivalent doses. Amla powder is available from Indian grocery stores and online health retailers across Britain and North America. It has a sharp, astringent flavor that works in smoothies, mixed with honey, or taken in capsule form. 5,000 years of use, the highest antioxidant score ever recorded, and it costs a fraction of what any superfood supplement commands on a health food store shelf.
Schisandra. Soviet cosmonauts were given Schisandra preparations before space missions. Soviet Olympic athletes took it during training. The Soviet government funded research on it from the 1950s through the 1980s. This was institutional science, not folk medicine. Schisandra chinensis is a woody vine native to northeastern China and far eastern Russia. It produces clusters of small red berries containing compounds called schisandrins. Chinese medicine classified it as an adaptogen, a substance that helps the body maintain equilibrium under physical and mental stress, and used it for over 2,000 years. The Soviet research focused on performance and endurance, but modern research has looked at Schisandra more specifically for aging-related applications. Studies have found that schisandrins protect liver cells from oxidative damage, the liver being the organ most directly responsible for clearing the metabolic waste products that accumulate with age. Separately, research has found that schisandrin B, one of the specific compounds in the berries, has neuroprotective effects, reducing oxidative damage in brain tissue. A 2014 study in phytomedicine found improvements in cognitive function in aging mice given schisandrin B. The skin benefits come from anti-inflammatory activity that reduces the chronic low-grade inflammation now understood to be a primary driver of visible aging. One plant documented effects on liver, brain, and skin aging simultaneously. 2,000 years of use in Chinese medicine, government-funded Soviet science, and modern pharmacological research confirming the mechanisms. Schisandra berry powder and supplements are available from specialist health retailers. The berries themselves, genuinely unusual in flavor, all 5 tastes present simultaneously, can be grown in temperate gardens across Britain and North America. Gotu Cola. Gotu Cola has been called the herb of longevity in Ayurvedic and traditional Chinese medicine for 3,000 years. In Sri Lanka, it is said that elephants, among the longest-lived land animals, eat it regularly. Whether or not that explains elephant longevity, the compounds in Gotu Cola have now been studied extensively. Centella asiatica is a low-growing plant found across tropical and subtropical Asia. The active compounds, asiaticoside, maticoside, and asiatic acid, have attracted significant research attention for their effects on collagen synthesis and wound healing. Collagen is the structural protein that gives skin its firmness and elasticity. Production declines with age, beginning in the mid-20s and continuing steadily.
A 2012 study in the Journal of Ethnopharmacology found that Gotu Cola extract significantly stimulated collagen synthesis in human skin cells. This is not a cosmetic claim, it is a measured, documented cellular response. The same compounds have been studied for effects on the brain. A 2016 study in the Journal of Alzheimer's Disease found that Asiatic acid reduced amyloid plaque formation, one of the hallmarks of Alzheimer's, in laboratory models. Earlier human studies found improvements in memory and mood in older adults taking Gotu Cola extract for 2 months. Gotu Cola is eaten as a fresh vegetable across Sri Lanka, Indonesia, and parts of India. It is added to salads, juiced, and made into drinks. In the West, it is sold primarily as a supplement. 3,000 years as a longevity herb, clinical research on collagen, brain protection, and memory, and a growing body of evidence that the traditional classification was not wrong.
Jiaogulan. In the Guizhou province of southern China, there is a region with one of the highest concentrations of people over 100 years old ever documented outside of official blue zones. Researchers investigating the population found one consistent habit. Almost everyone drank Jiaogulan tea daily. Gynostoma pentaphyllum is a climbing vine in the cucumber family. It is not related to ginseng, but it contains compounds called gypenosides that are structurally similar to the ginsenosides in ginseng, the compounds responsible for ginseng's adaptogenic properties. Some gypenosides are identical to ginsenosides. Jiaogulan has been called poor man's ginseng because it grows abundantly as a weed in southern China, while true ginseng commands extraordinary prices. Japanese researchers discovered Jiaogulan accidentally in the 1970s while surveying the region's unusually long-lived population. Their subsequent research found that gypenosides activate AMPK, adenosine monophosphate activated protein kinase, an enzyme sometimes called the metabolic master switch. AMPK activation is one of the mechanisms through which caloric restriction extends lifespan in animal studies. It is also the mechanism targeted by metformin, the diabetes drug now being studied specifically for anti-aging effects. A 2013 study in the journal PLOS One found that Jiaogulan extract extended the lifespan of the nematode C. elegans, the standard laboratory model for aging research, by 24%. Animal studies are not human studies, but AMPK activation through Jiaogulan has been replicated across multiple research groups. Jiaogulan tea is inexpensive, available from Chinese herbal suppliers and online retailers across Britain and North America. The plant grows easily in temperate gardens as a climbing annual. A village of centenarians, a mechanism that matches what pharmaceutical anti-aging research is targeting, and a tea that costs almost nothing. Ashwagandha. Cortisol is the stress hormone. It is also one of the primary drivers of accelerated aging. Chronically elevated cortisol damages brain tissue, breaks down muscle, disrupts sleep, accelerates skin aging, and suppresses immune function. A 2012 randomized controlled trial found that ashwagandha root extract reduced cortisol levels by an average of 27.9% over 60 days. Withania somnifera has been used in Ayurvedic medicine for over 3,000 years as a rasayana, a rejuvenating tonic. The name ashwagandha means smell of horse in Sanskrit, a reference to the root's aroma and to the traditional belief that it confers the strength and vitality of a horse. The 2012 study was published in the Indian Journal of Psychological Medicine. It was a double-blind, randomized, placebo-controlled trial, the gold standard of clinical research. 300 milligrams of ashwagandha root extract twice daily for 60 days. Cortisol reduced by nearly 28%.
perceived stress reduced by 44%, sleep quality improved significantly. Subsequent research has looked at ashwagandha for testosterone levels in men, for muscle recovery after exercise, and for thyroid function. A 2015 study in the Journal of the International Society of Sports Nutrition found significant improvements in muscle strength and recovery in resistance-trained adults taking ashwagandha compared to placebo. The mechanism connects directly to aging. Lower cortisol means less tissue breakdown, better sleep, reduced inflammation, and improved immune function, four of the primary factors that determine the rate of biological aging. Ashwagandha supplements are widely available. The clinical evidence behind them is more robust than almost any other herbal supplement currently on the market. 3,000 years of traditional use and a 2012 controlled trial pointing in exactly the same direction.
Astragalus. Every cell in the human body has a biological clock. It is called a telomere, a protective cap at the end of each chromosome. Every time a cell divides, the telomere shortens. When it runs out, the cell can no longer divide. That process is one of the primary mechanisms of aging. In 2009, the Nobel Prize in Physiology or Medicine was awarded for the discovery of how telomeres protect chromosomes and the enzyme that maintains them. That enzyme is called telomerase, and astragalus is the only plant ever shown in peer-reviewed research to activate it in human cells. Astragalus membranaceus has been used in traditional Chinese medicine for over 2,000 years as a tonic herb, something taken regularly for general vitality and longevity. It is one of the most widely used herbs in Chinese medicine and has been for centuries. In 2009, a study published in the journal Rejuvenation Research found that a compound extracted from astragalus, called cycloestrogenol, activated telomerase in human T cells and CD4 cells. The implication was significant enough that a pharmaceutical company commercialized a purified version of the compound under the name TA65, sold at extraordinary prices as an anti-aging supplement. The research behind cycloestrogenol and telomerase activation is real and peer-reviewed. Whether telomere lengthening in specific cell types translates to meaningful anti-aging effects in whole humans is still being studied. The biology is genuinely complex, but the mechanism, activation of the enzyme the Nobel Committee identified as central to cellular aging, has been documented in human cells from a plant compound. Astragalus root is available as a dried herb, powder, and supplement from Chinese herbal suppliers and health food stores across Britain and North America. It has been used continuously for 2,000 years. The modern research identified a mechanism that may explain why. Every other plant on this list has impressive evidence behind it. Astragalus is the only one where a specific compound has been shown to interact directly with the cellular mechanism of aging itself. 2,000 years of use as a longevity herb, a Nobel Prize-winning mechanism, and a peer-reviewed study showing activation of the relevant enzyme from a plant compound. That combination does not exist for anything else in this video.